motor neuron gene expression program. And actually Tulsi was able to use other tools that I probably should not really get into here, but she was able to identify what families of transcription factors are controlling the adult state and the embryonic state. And remarkably, while she found that the adult state is controlled often by neuronal activity, which means firing action potentials, as well as exposure to hormones, different hormones in a maturing organism. The juvenile program was controlled by actually our well-known factors ILAT1 and LHX3 that define the identity of motor neurons early on. These transcription factors, so I already showed you that these two transcription factors are sufficient to convert embryonic stem cells into motor neurons, but remarkably, very soon after motor neurons become born in the embryo, these factors are downregulated. So the cells actually stops producing these transcription factors, and then the cell goes through this maturation and aging state when the motor neurons become susceptible to the mutations that cause ALS. And so we actually asked a relatively straightforward question: what would happen if we re-express the factors in the adult motor neurons? Would we be able to actually change the state of these cells and reactivate some of the more immature properties in these cells that would make them more resistant to the mutations and the accumulation of misfolded proteins in these cells? But this is not, it's very easy to write it on a blackboard, but it's not actually that easy to do it in biology. So actually what you have to do is you have to start by isolating the ILAT and LHX3 genes. So you have to find the stretches of the DNA. You have to usually put them in bacteria. And then we have to actually change the regulatory sequences that control in which cells the genes are turned on. Because these genes are turned on in adult motor neurons, so we have to find other regulatory sequences that would allow these genes to be turned on in adult motor neurons. And then how to get them into adult motor neurons, we have to package them actually into viral particles. We used virus, which is called RNA-associated virus, that actually if you inject into animals, it will infect very effectively most of the neurons. And then after we infect these mice with the viruses, we have to check that the genes are turned on in motor neurons, and very selectively in motor neurons, because we did not want to reprogram other neurons into motor neurons. That could be quite bad. And so here is the viruses that we produced, and we injected them actually in a newborn mice. And then here you can see again, this is a cross-section of spinal cord, and now you see this is adult spinal cord. Normally you would not see any of these red and green dots, so each of these dots is actually nucleus, and the transcription factors go into the nucleus where they control gene expression. The blue delineates the area where motor neurons are found, so this is the ventral part of the spinal cord, and you can see that the transcription factors are highly expressed in the nuclei of these motor neurons after we infected these animals, suggesting that the tool that Tulsi developed worked really well. And so now with this in our hand, we wanted to know what happens if we now infect motor neurons in a mouse that carries the mutation that causes motor neuron degeneration. And so we used a mouse model of ALS that actually expresses the SOD1 protein that I said is mutated, was found in one of the first patients. So this mouse expresses the mutant SOD1 protein. And normally in this mouse, you start seeing aggregation of misfolded proteins already on postnatal day 45, and then you start seeing actually loss of motor neurons and behavioral deficits, so the mouse stops moving by postnatal day 90, and at 150 days the mouse dies, while normal mice live for 2 years. And so in the first set of experiments, we injected the virus at postnatal day 1 and then asked whether we can see any changes in these misfolded proteins. And so this is now looking very high magnification. So this is a cell body of one motor neuron. This is the nucleus. This would be another motor neuron, another motor neurons. Now this is a healthy motor neurons, and the green staining is for actually structures that degrade misfolded proteins. And in healthy cells, you see these little small dots that are each basically degrading misfolded proteins. But in the case of ALS, you start seeing these pathogenic large aggregates that accumulate inside the cytoplasm of these motor neurons. So when we then added the virus, so we added first virus that expresses control genes. In this case, it was red fluorescent protein. And you see that still these motor neurons that express the red fluorescent protein have these large aggregates of misfolded proteins in them. But remarkably, when we now express islet 1 and LNChex3 shown in red, so again these factors, because they are transcription factors, they go into the nucleus where they bind to DNA, and they change the gene expression program in these cells in such a way that these green aggregates look much more like the aggregates that you found in a healthy organism. So that to us really suggested that we were able to make these motor neurons somehow capable to handle the mutant proteins as well as young motor neurons would. And so this gives us a hope that one day we might be able to find ways how to actually rejuvenate adult motor neurons and maybe help them to fight the mutations that cause degenerative diseases, not only in this case that would drive from ALS, but in other neurodegenerative diseases. So this brings me to the end of my presentation, and I hope that you will be able to take away a few messages that if you think about why motor neurons are susceptible to the disease, not other types of neurons, it might be because motor neurons are more sensitive to accumulation of misfolded proteins, and that we identified actually a pathway that is called MAP kinase pathway that these cells activate that leads to their degeneration. And the drug that we developed together with Brent Stockwell inhibits this cascade, this pathway, the signaling pathway inside the cell, and prevents or stops motor neuron degeneration. And then in the second part, I hope that I was able to show you that motor neurons undergo this very prolonged maturation where their gene expression program changes. In the mouse, it takes 1 month of their life, which in human it could be easily equivalent to until you become a teenager. And then during this process, the motor neurons become less capable to handle misfolded proteins, and you start seeing an accumulation of misfolded proteins as soon as the motor neurons reach the mature age. And then we have now ways how to actually change the age status of these motor neurons, how to reprogram them into more immature state, or state in which they are more resistant and they can clear the misfolded proteins. I think I acknowledge most of the people that contributed to this work, but Emily Lowry, John Costa, Esteban Mazzoni, Tulsi Patel, Stefan Nedelek, and Sebastian Tams were the main people from my lab that generated all of this wonderful data. And I'll be happy to answer any of your questions if you have. Thank you.